Yeah, is the screen visible now?
Yes, it's visible.
Okay. So our next session is titled Applications of Chemometrics for Quality by Design in Pharmaceutical Industry by Ms. Rucha Deshpande. Ms. Rucha is the founder and partner at Let's Excel Analytic Solutions for promoting data science in research. The startup in India recognized startup that offers software as a service platform for data analytics. The startup is engaged in developing machine learning and artificial intelligence platforms that can be used for breakthrough research in science and technology. An independent trainer and consultant for Six Sigma, Lean Statistics, Machine Learning, PAT, or Chemometrics. She is a chemical technologist specialized in pharmaceutical sciences. She has experience in pharmaceutical product development in accordance to quality by design principles and is also a science writer. With this short introduction, I will now request Rucha to kindly start the session.
Hello, everyone. Am I audible?
Yes, Rucha.
Yes,
you can start sharing the screen.
Yeah, sure. First of all, thank you so much, Venture Center, for inviting me here. It is such an honor and a privilege to address a learned audience like this. And especially because Chemometrics is close to my heart as our startup Let's Excel Analytics Solutions is also doing its bit for this for Chemometrics. Sorry, I just realized I have not shared my screen. I'll just share my screen. Okay. So as we run through the slides, you'll get to see what exactly our startup is doing for Chemometrics. Our idea is to make Chemometrics available to everyone. It should be as simple as browsing through the web pages of social media handles. Like probably when we browse through Facebook or Twitter, we don't consider what we are doing, and we are just able to move across the pages effortlessly, and things are very simple. So our objective is exactly that. Although we don't say that we just want to limit ourselves to chemometrics, data analysis in general is something that we want to make available to the masses. So this is our objective, and we will see as we move through the slides how we are contributing to it. Because my primary experience is in pharma, I have limited the topic. Means the title says that we are going to discuss chemometrics for quality by design in pharmaceutical industry. But whatever I'm going to speak is very much relevant to other industries, and the other industries were in fact doing it better than the pharma industries a decade ago. So this should not be something which is not known or which is not practiced in other chemical industries. So don't worry if you are not from pharma industry, this is still relevant to you. Can you see my slides? I would just request a confirmation.
Yes, ma'am.
Yeah, thank you so much. So the contents that we are going to cover today are, like, first we are going to talk about quality by design and how we can achieve quality by design using chemometrics. Then we are going to take some application-based case studies to find out how we can use chemometrics for vendor selection or for raw material selection or reverse engineering and PAT. Generally in pharma industry, it's a belief that for chemometrics, we need to have data which is coming from spectroscopic instruments. So to break that norm, I'm also going to include some case studies which are not from, which are run on non-spectroscopic data. And already you have seen in Dr. Keshav's presentation, he also used this particular means the case study which he showed was non-spectroscopic in nature. So when spectroscopic data is there, PAT is there. And this is what I means when I learned chemometrics, I learned it for the PAT because 10 years back I was also in Dr. Reddy's like Dr. Keshav. And at that time, our team was developing these chemometric models for real-time process controls. So that is something which is very much used in pharma industry, and it's called as process analytical technology, which is nothing but real-time process control methodology. Means if you um, have a real-time manufacturing process, you just know where is the end point using chemometrics, and you stop the process there. And if your process is not progressing as it should progress, you know that it is not progressing like it should progress. That's why you stop the process. So this is what PAT is for non-pharma audience. So first thing first, what is quality by design? If you ask me quality by design, I would say what is quality by design? I would say that it is beginning with the end in mind. So why do I say that? Because if we want to understand what is quality by design, first thing that we should know is what is not quality by design. So if you look at this part of the slide, which I am circling, is related to what is not quality by design. If something is not quality by design, I call it quality by accident or quality by chance. So you can think of more such things, and that would keep you alert during the session. So and just message them in the box, because I know many of you have come across processes where quality by accident has happened to you. So such incidents and such words, I love using them. So if you have some creative words for not having a process which is not quality by design, just text them in the chat box. And during the session, if you feel that I'm not being clear somewhere, feel free to interrupt me and ask me questions, because it is better if we clear your doubts right there. So coming back to what is not quality by design. So in old days, like Dr. Keshav was mentioning, in the old days, and when the before the Industry 4 revolution happened, what we used to do is we used to buy input material from whichever vendor we had access to. That input material used to be variable in nature. Then we had fixed processes. Specifically in pharma industry, this was a problem because I see couple of colleagues from pharma industry, and I know that if you'll ask them to change their method of analysis, method of manufacturing or anything, they will raise their eyebrows because they have to follow a very lengthy and complicated processes of getting their change controls approved and giving justifications. So what we used to do is we used to just have a normal fixed process. We never used to touch it. We never bothered about what is going to be the output. If the output meets your quality objective, like there was a chance that you probably could meet your quality objective. And if you made your quality objective by chance, you would then only sell your product. So this is very commercial outlook which I'm trying to give to you all. Now, what I'm trying to say is, if by chance your product met the desired quality, you were selling it. Imagine that you are going to a pharmacy store and you are buying a tablet, and that tablet doesn't have the active pharmaceutical ingredient, which should be there between 98 to 102%. Would you like that pharma company? Would you like such thing to happen to you? So obviously no. But this used to happen, And the people used to, depending on the regulatory norms in those countries, you would get variable output. And if you don't meet that, people used to just throw those batches. So if you throw your batch like this, you're going to waste a lot of material. So if you're wasting your material, you're wasting your cost that went into production. You wasted your money, which went into inspection. You wasted your money, which went into testing of that batch. A lot of money, right? So wasting this much of money doesn't make sense when you are involved in a commercial business. If you're doing this, you need to stop doing this right now and start using techniques which will help you have efficient control strategy. So how do we do that? We do it through quality by design. And how we do is, in quality by design, we are laser focused on defining output quality. So for example, if I want my active pharmaceutical ingredient to be present between 98% to 102%, I will define it right before I start my project. And in quality by terminology, we also call this as QTPP. So for non-pharma attendees, QTPP is nothing but your finished product specification. So basically, you're laser focused on your finished product specification. Then what you're doing is you are designing a process to meet that output quality. So even though if you see this arrow like this, actually you're beginning with the end and you're actually designing your process to meet this product quality. And to meet that product quality, you are following a control strategy. Now, in this case, I'm suggesting that there are various methods of having your control strategy in place, and chemometrics is one of them. So we'll see how chemometrics can help you in quality by design. So because you need to have an efficient control strategy, you need to know how things are going to work in advance, how they're going to work in advance. How would you know that? Even before you start your experimentation, how you're going to know that how things are going to work in advance. So to know this, you need to create a knowledge base. So if you're in generic industry, you already have innovators. So maybe what you will do is you will get your innovator, you will do advanced characterization on the innovator, and then you will try to rework on how things can work. You can hypothesize few things and find out by using the relevant data. One is literature, one is your experience in previous similar products, or the other way is to generate the data yourself. So this is really not a magic wand. To make chemometrics work for you, you have to do a lot of hard work. You have to generate the data yourself. And it is not one person's job. This is because from 10 years I'm doing this. I can say that even if it feels like sitting on a computer and building nice models, this is really not one person's job. It requires a lot of cross-functional working. So to create this knowledge base, if you don't have that knowledge base in literature, you actually use advanced characterization techniques. So like VC Analytics has many advanced characterization facilities with them. Probably that's why they're interested in chemometrics. And many pharma companies, not just pharma companies, other chemical industries have used these advanced characterization techniques, which can include like DSE or maybe LCMS, or like these regular like NIR, Raman, hyperspectral imaging, which is an extension to this. So all these advanced characterization techniques you can use to analyze the data using chemometrics. So in the end, this is the last step you're going to do. This is when your software or something comes in picture where you are analyzing. So before all this happens, you have to do a lot of brain work and a lot of data generation, which which is not generally spoken of. So after you analyze, you can build a predictive model. You can start predicting future outcomes using your predictive model. And if the model is like well validated, and if you are confident that it serves your purpose, then you implement quality control mechanism in at a manufacturing scale. So this is how it works. To summarize, I have this workflow for you all. So first thing is, we need to have a business problem. If we don't have a business problem, we are not going to create a value out of that chemometrics project. Because I come from the Six Sigma background also, I generally prefer to have a business value. So what is your business problem? What are you trying to solve? Is there a specific legacy product in your organization, or the product you are developing for your client, which is consistently failing on certain criteria? Let's say, for example, if is it a polymorphic transformation? If your API is undergoing polymorphic transformation, it's unstable, and because of that, you're selling your product to someone, and that company or industry is rejecting your product. If that is happening quite often and resulting in losses, then this is a business problem for you. What do you do after you have a business problem? After you have a business problem, you hypothesize. Okay, I have problem with the polymorphic transformation. Now probably I need XRD data, or maybe I need some other kind of data which will show that polymorphic transformation. I'll generate that data on my positive control batches and negative control batches. So when we say positive control batches, these are batches which work very well, which were not rejected by your client. And you will have them at one place. You will have your rejected batches at one place. You'll acquire that data. You will remove some data which is irrelevant. Because, if you're going to, as I said, you may not have all the technical capabilities. You may have to borrow some technical skills from somebody who is more experienced in that area. Like, for example, in my case, I'm not an advanced characterization expert. So if somebody gives me data, I would just say, okay, give me raw data, but I wouldn't know how to process that data. So this is something which should come from advanced characterization expert who knows, okay, this is garbage. We don't want to give this to Rucha. We'll just take it out. So remove that garbage. And then next is transform and visualize. So transformation is required in some cases. It may not be required in some cases. Visualization is just a first step which you go through to have a feel of the data, how the data looks like, and how whether it is usable or not usable. And then next is you build your model, validate your model. Once your model is ready, you report it and you deploy it in your manufacturing facility. Now, the next time polymorphic transformation happens, you get an alert. And with that alert, you stop processing. And because you stop processing, you save a lot of money, which would have gone into further processing of that batch. So this is the concept. Now we will see few examples here. Now this is a hypothetical example which I have created. After this, we will see one actual casework which is being done at St. John's University, and that is all in progress. And at a later part, we will have a PAT application if the time permits. So speaking about, I think I'll just, before we jump, I think we should have a quick check if everybody is on the same page. So I'll ask a small question. The question is, what is the first step in chemometric workflow, which we just spoke of? There could be multiple, if you're not following quality by design framework, there is a reverse approach also, but the approach which we spoke of If somebody could type in the chat box, I'll be assured that you all are listening. Come on, guys, this is workshop and we should be in alert mode and it should be two-way interaction. Please, if somebody, yeah, I see someone responding. What is the end result quality? Okay, yeah, this is good one because yeah, it comes from this slide that we are actually start beginning with the end and it's the what exactly we need. So if we are wanting to make tablet formulation, we don't want it to be brittle. It should be so and so color. It should be so and so size. It should have so and so embossing. This all comes in finished quality specification for that product. Finished product quality specification. So this should be one. So we just have one answer. So this is for quality by design, but this is not the answer for what should be first step in starting your chemometric workflow. Yeah, problem definition, business problem, right. So we have a business problem and then we hypothesize that if we have so and so data and we analyze that data, we will get our answer. So this is generally the first step. Means we first have a business problem and then we say that, okay, I have this kind of data and if I analyze this, I might get the answer. So this is the first step. Now let's go to our case study. Thank you so much for your response, guys. It also keeps me interested in telling you more things. So let's go to our first application. Application in vendor selection. So what I'm going to do is I will show you the first slide and then we'll actually go to my uh, we'll actually go to the software which we have developed in Let's Excel Analytics Solutions and we will analyze this data in the software itself. So the problem statement here is there is means we are buying several APIs from a vendor and that vendor is supplying us APIs with variable thermal qualities. So in pharmaceutical industries, why this thermal quality of API is more important? Because let's say you're going to process that data, sorry, not the data, always the data is on my head. So let's say if you're going to process your API with a technique called hot melt extrusion, which is used in other industries as well, plastics and similar chemical industries. So if you are going to subject it to a lot of heat, what you will end up getting is degraded form of your active pharmaceutical ingredient, which will not have desired therapeutic effect inside your body. So that is the reason we need to ensure that the API should be thermally stable. And to find out that, so this is a problem that we are buying our product from a vendor with a variable quality. And now we want to be assured that we just select the vendor who can supply us consistent quality of API. So in this case, we have an hypothesis. Like one of our scientists who is here went through some research article, which is here. Means this is the data source. You have this data. It is available free of cost. And then he goes through this data. He realizes that there is a nice DSC data in this article, and probably we can use this data to build a model which can predict the thermal stability of APIs. So we think that, okay, nice hypothesis. Let's just give it a try. So right now I have stored this data on my desktop. So I'm just going to take it from there. Before we jump to data, these are the uh, properties of ingredients, so not the ingredients, the APIs which we are going to study. That's the glass transition temperature, onset temperature of melting, onset temperature of crystallization, enthalpy of melting, Gibbs free energy of crystallization, entropy of melting, molecular weight. Those who are really good at chemical engineering, they would be very much familiar with all this. Those who are handling DSC data would know this much better than what I'm saying now. So let's go to the data. So the idea is not to reconfirm that the science works. We know that science, there is a lot of science behind this data, and there is a lot of science already there in place which can tell us what can happen if API is stable and what can not happen when a stable API is not stable. But we don't know what is the threshold where we should stop. What should be the TG where, for declaring that this API is stable. So for that reason, to draw that boundary line, we will go and analyze our data. So I'll just stop presenting and we will so what we'll do, we will go to the website where we host our applications. Many of you might be already using this, but for those of you who are not using this, these are available free of cost for access. So you can, if you want to request access, this is our website. You can go here and click on request free access, fill up the form, and within a week we will give you access credentials. So the reason we are giving it out like this is because we want to make data analysis as simple as browsing social media pages. So let's go here. We will analyze this data with PCA. As Keshav said, this is really a magic tool and this is really a preferred tool of choice for analyzing most of the data sets. So we'll go to this PCA application. When you request access, you'll get the username and password for accessing. I do have so many passwords that I forgot my own. Okay. Yeah. So luckily I remember it. Now let's go. Here is the data. So this is known sample data. I'll just import it. The data is in CSV format. There are some guidelines for handling this data and some other information which may be useful to you. You can find that information here. So I'll just for time being, I'll erase everything. I like to highlight a lot because I think because when I am in presentations many a times I don't understand what is happening. So that's why I'm just looking for these highlight tools all the time. So yeah, this is the data. And as you see, this data has like these are all your APIs. These are all the properties which we saw here in the slide. So these are all the properties which are listed here once you imported data. And this is the experience based understanding, which says that which one out of these means out of all this, some are stable and some are unstable. So in your default view, it will only show you 10 samples. Now we have 24 entries here. So we can extend the view. And now you can see that it shows all your samples. Now in all the samples, you have some samples which are labeled as yes, some samples which are labeled as no. This is based on the experience of several batches, let's say. Means if you're trying to do this for your own company or own industry, there you will see that you know what works and what doesn't work. So based on your experience, you have said that, okay, some of them are stable and some of them are unstable. Now, the next step that you need to do is very important because if you don't do it, you will get series of errors here. So to not get these errors, I would suggest that you first select the categorical variable. Categorical variable is nothing but the data which is labeled in the form of yes or no. So if you speak of data types, there are two types of data types. One is numerical data type, the other one is categorical data type. In numerical data type, you have two types again. One is the discrete data, and the other one is continuous data. The discrete data can be in the format of 1, 2, 3, 4. It will never have decimal points. But if you're talking about continuous data, you're talking about 1.1, 1.12, like this. And if you're talking about categorical data, there again you have two types. One is ordinal, the other one is nominal. So in the ordinal data, you will have order in the data. Like we have high, low, medium. And if you're talking about nominal data, you will have labels like male, female. So this is about the basics. This is the basic concept in statistics, basic terminology which is used in statistics. So that is the reason you will see this label categorical data. You need to select it, because the reason you need to select it, you need to differentiate it from the numerical data so that the software will know that what data it needs to process. Now you move on here for the correlation matrix. It still shows error has occurred. There must be some reason. So we'll dig down. Now we come here in the data summary, and here we get to see that we have some NS. NS is not available. It's a missing value. So how do we take care of that value? We simply, okay, we simply go here and replace it with approximated PCA decomposition. Now your NA values got missing. Yeah, so that's what our purpose was. The algorithm has replaced the values with approximated values by itself. So you can either keep them or you can just remove those, because these were just three values. Depending on your case, you can either remove them or you can go with this processing here. I chose not to remove them because we already have very few samples here. And this software is capable of handling really huge amounts of data. Like a few weeks back, I almost analyzed 1 lakh women's data. So it has a huge capacity. So if you have really large data, feel free to explore this. This is not a spectroscopic data, so I'm unchecking that. If I check this, I'll get this plot, which I don't need at the moment. So I'll just uncheck this. And I will do the data pre-processing, the standardization, which Dr. Keshav explained in his slides. So why we are doing the standardization? So I'll show you visually what happens when we do standardization and what happens if we don't. So here is your raw data. And what you do is, now you see that this specific variable, molecular weight, it is having higher values as compared to the other samples, other variables, I would say. So what we need to do is we need to bring it on to the same level as the other ones, because unlike other techniques, this technique depends a lot on your variance. If you will have higher variance, that means you will make your model biased towards that specific variable. So your model can get biased towards molecular weight, and you will think that molecular weight has the highest influence among all these variables. So for not getting biased, what we are going to do is we are going to go ahead with data processing. So we are just going to, this is a general practice. This is the basic minimum data processing that we do. So we are going to just do mean, center, and the scaling. And then now you see that they're almost on same level. It won't happen like a perfect case. It is not going to happen like all will be identical, exactly same. Some, these are natural things. This random variation, minimum random variation is expected, and we need to live with it. So without now wasting much time, we are ready with the data. We are ready with our um, samples. Now we will just go and see how our PCA plots look like. You just have to do these 2-3 things. Simply look for data abnormality like we did. We just removed some missing values. then we mean centered, then we standardized. Now it's time that your PC is already available. So here you will see all the calculations for your eigenvalues and your explained variance and cumulative. So this is cumulative explained variance, means what is the, sorry, I highlighted wrong field. This is cumulative explained variance. So this is what I prefer to look at, means if you're doing data analysis, don't get bugged by too many things. Few things which are important to know are how much the variance is getting explained by your sample. So we will have a quick revision of the theory, means um, if somebody needs it, they can just have a look at what I mean by explained variance if you have forgotten. Although Dr. Keshav has explained it very well, I like to show this graphic very often. So if you see, this is the scatter of the data, and this is how it is spread up. Then you draw an imaginary line like this, which is here in this plot. And that imaginary line rotates like this in three-dimensional space. So when it rotates, it draws some projections on itself from those data points. And in the direction of those projections, in the direction where you have largest variance, that line will stop first. So the largest variance is in this direction, then in this direction, like this. So it will go on, and this first line will be your PC1. The second line will be your PC2, and so on. So I'm sorry, is there some noise at my background, or am I audible?
So we can hear you clearly.
Yeah, okay. So, so this is how we give importance to the eigenvalues, means they are arranged in the descending order of importance. So that is why PC1 shows you the largest information, then PC2, and then PC3. And eigenvalues is nothing but the scalar, which is related to the vector. Yeah, I'll just show. Yeah. So eigenvalue is nothing but the scalar which is related to the vector of your principal component. So principal component is a vector. It has a specific direction. It is not just a line, it has a direction. So that is why, let's say this is your direction. So the scalar which can be used to multiply to this vector is your eigenvalue. So this is the very layman's explanation, I would say. I'm not, since I have learned chemometrics on job, and I have not gone to some colleges. By the way, when we're speaking about colleges, I would recommend ISI India, which has many courses, advanced analytics courses, and they do cover these techniques like PCA, PLS. They may not cover it on chemical data, but these techniques are very much prevalent in the statistical world, and people use them um, in other industries. And that is why, if you want to go and attend a course and have a very technical hold of all the terminologies which are used in principal component analysis, I would suggest that go for Indian Institute of Statistical, um, I don't remember, Indian Statistical Institute, ISI. It has branches in Mumbai, Delhi, Kolkata, and Hyderabad. So we'll clear this and we'll just go to our explanation. So that was tabular data, which was talking about the same thing. It may look complicated, but it's not that complicated. Now we'll go to PCA summary plot. So what we see here is something which is not clear to explain, but if you just have some sample names here, you can see. And I think this plot it has probably very small labels, so it may not be very much visible. Don't worry if it's not visible. We'll go here and see individual plots. So don't worry about that. Basically, let me explain some concepts before we move on to the next slide. So this plot shows how each variable is related to every sample in your data set. So if we are talking about, let's say, phenidipine. So phenidipine is towards this side of your plot, it's the bottom right side. And which is the property which is closer to the bottom right side? It's molecular weight. So that's when probably phenidipine is, the location of phenidipine is here because of the molecular weight of phenidipine. Similarly, you see some APIs which are here at the top portion of your plot. Probably their property values are high for these samples. And we are going to see this in next plot, where you will see this in much clear representation. This is just a summary plot, and we have various plots here which will make your work easy. So this is the same scores plot. This is the same plot which is here. It's again here, just that you have this probability circles here. I'm keeping them as it is for right now because the purpose is just to explain things. So now here you will see that we have two groups of samples. One is yes and the other one is no. So this information comes from your data set itself. Remember, in the beginning of data import process, we labeled some data as yes and no. So this information comes from there, and it groups the data into two different colors. We see a lot of overlap here, which means in these two circles, means these circles are broadly differentiating your data into the stable and unstable APIs. So we see a lot of overlap. What it means is it may not be possible to perfectly separate these two categories. What could be the accuracy with which we can differentiate these two categories? That too we are going to see in this example. So as of now, stay tuned. Just stay focused on our objective. Our objective is to find out if the vendor can supply us the stable thermal API or not. So I'll just clear all drawings. Just as I said, this one, for example, albendazole, I'm highlighting it. So its location is here. And now I'm interested to see what is that which is causing it to be here. See, I have highlighted that area, and you can now see in the loadings plot, these three properties are related to the reason because of which albendazole is located in this portion of the plot. So this is how we find out cause relationships. So this is just one part of it. There is a lot that we can do with this data. We'll explore that. Now from here, if this information is not clear and if we don't like these plots, we can move on, and we have another view which is called as biplot. So if we go here things are very much clear. If somebody out of you is familiar with R, you would know that all of these are nothing but compilations of some R packages, which we have done and made available free of cost for you all, so that you can do your data analysis at your end. And if there are any advanced requirements, you can always reach out to us so that we can do all those advanced modeling part or advanced software related aspects which you need to be incorporated in the form of features or customizations for you. So this is the biplot, and what the information which biplot gives is similar to the scores and loadings plot, because this is nothing but scores and loadings plot together. As you can see here, we have two classes again, but in this case, again, we see that these three properties, which we had seen earlier, they're again closely related to each other, and they are pointing towards a specific direction, towards this sample and this sample, which means these two samples are likely to have high values for this property, whereas this sample is likely to have low value for this property. So this is how we interpret biplots. Similarly, this sample is going to have low values for all these properties. So this is very much directional, very intuitive. There's nothing much we need to do. We just need to look at the direction in which those those arrows, or you can say the vectors, are pointing, and that can help us decide, oh, what is causing the placement of this particular sample in this newly designed space. So this is about scores, loadings, and one important thing that we missed on is the explain variance plot. So the elbow point, how many principal components we should have for building a model. If we are using these two data sets, means if we are building a combined model for stable as well as unstable APIs. So in this case, the thumb rule is that we stop at the elbow point. So here we have the elbow point. So beyond this point, if you will add this, here itself we have about 70% of information. And if we add another 15%, that would be close to, this is 74 plus 15, which will give us about 89% of information. And beyond this, there is not much change or addition of information. So less complicated models give us a more robust output. So we can say that we can stop at the third principal component for building a combined model for these two data sets. Now we will move on to the next step. I just got data. Okay. So here, this was about principal component analysis. Now we are going to find out how we can build separate classification model for thermally stable APIs and thermally unstable APIs using SIMCA methodology. So if you will jump onto this page, initially you will see there is some error, but it's not an error. You simply need to select the class for which you're going to build the model. So in this case, let us build the model for unstable APIs first. To build that model, what we are going to do is we are going to select the group of principal component, group of those samples which we'll use for making that individual principal components. So as Dr. Keshav has rightly explained, SIMCA is nothing but extension of principal component analysis. So you simply go here, you decide how many number of principal components you want. Using this knob, you can change. So for example, now I change the number of principal components to 2. When I change it to 2, I see that explain variance is 84. Now I change it to 3 and my explain variance has gone high. So this is how dynamically you can evaluate your models. If you'll go for some other software, you might have to build a model with 2 and then write down the values in some Excel sheet and then save that model. Then again you have to go and again rebuild a model with 3 principal components. Okay, I got 97% of cumulative explain variance, then write down the value, and like that you might need to choose. But in our case, it's very simple. You simply change here, you will get the value reflected in this box. So now we will clear this. Okay, I've chosen these three samples. I'll go here. I'll save my model file. If you're using this software already, I would say that start using it as soon as, if you have access, I would say that start using it as soon as possible, because at later point, we might just disable these functions of saving models, uploading models, and they may be available only for premium users. So right now, if you're using start using them as soon as possible to make best use of it. Now, second is just like we built it for unstable APIs, now we are building it for stable APIs. Now we have selected yes here. Earlier it was no. So we will go down and then we will just save the file again. So we have saved 2 files. Now we need to upload these models. So I have uploaded, I have already saved them somewhere. So I'll just go and use the same files because I don't have to find them in the download section. Yeah. So something strange has happened. Probably I selected wrong model file. Okay. Go again. So I have to go to downloads. This is what I don't like because I have so many files. Okay. So this is the file which is saved here right now. And we are using the same file to make predictions here. Now, when we upload this file, either you have a choice to see this data in this tabular form, or you can see it pictorially like I prefer. So these are again tabular forms. This is for train, this is for test. So one important thing which I missed is for validating this model, we need to differentiate our first data set. Like we have about 25 samples here. So we need to partition this data in train set and test set so that we can build a model using only train set and we can use the model to make predictions for the test set. So what this gives us? This gives us a confidence that, okay, the model which you have built can be used. It is not giving some random predictions. So to get that confidence, we are going to divide this, and by default means you don't have to do anything if you are new to all this. By default, it divides the samples into train set and test sets. Alternatively, if you want to include more samples in train and test, you can change this value and you can decide. Means depending on whether if you select 1 here, there will be 0 samples in the test set. And if you select 0.59, which is the default value for us, you will have about 40% samples in your test set and 60% samples in your train set. So this is our Simca. We go here. This is our predictions for train set and test set. So this is the same information which is there here in this tabular format. So don't worry much if it looks like boring. It looks very boring to you. This is a easier way of analyzing. Again, you see yes and no grouping here. So red samples are stable. So that means all these samples here are stable, which are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this 12 samples, it has classified as stable, and this 1, 2, 3, 1, 2, 3, 7 sample, it has classified as unstable. Now you see some odd man out here. This one is odd man out. This one is another odd man. What it indicates? It indicates that this was an unstable sample, but our model classified it as a stable sample. This is because of the overlap that we saw in this course plot. So, and this one is stable sample, but the model classified it as unstable sample. So this is a misclassification error with which we have to live with. So if you're buying the API, you can be more stringent about this, because even if you reject some batches out for some because of some doubt, it's okay, I'm rejecting some batches because I don't want to face future consequences of making a batch which is not meeting the end product quality specification. So in that case, it's safe to say that okay, I'll go with this kind of model. But if you're selling the API and if you really want to convince someone that you are like you're selling consistently the product which is meeting the quality specification which you're promising, then you need a more accurate model than this. So it depends on the purpose. It depends on your end goal, whether you will use this model or you will not use this model. So this is about how to analyze the Simca plots. So this was for trend set, and you will get a similar result for test set here. Here you will see that one of the sample, it could not classify either as yes or no. It has classified it as none. And this is how it's like, um, again, you have a misclassification error here for yes, actually. It's a stable API which is rejected as unstable. So this is how we can use these two models. And if you want to make future predictions, just for example, take it like this. You have the data here. This is unknown data, let's say. Now you don't have labels stable and unstable here. You simply have, again, you get the prediction plot. Only the difference is that you don't have the color coding here because you don't know what it is. Because this is supposed to be unknown data. You don't have to pay for DSC characterization once you have this model. So this is one advantage. If you don't have that instrument within your facility, you can give, let's say, uh, you can pay for initial model building part, and later on you can use this method as your in-process control for your organization. So for these many samples, the model said, okay, means for selecting the vendor, I said, okay, you are supplying stable quality of API. I'm going to buy API from you. And if it is like this, I'm simply going to say, sorry, boss, we cannot buy API from you. So this is how we can use this. API is active pharmaceutical ingredient. I've been saying API from long time. It's nothing but the actual drug molecule which gives you therapeutic effect inside your body. So this was about vendor selection. For next application, it is again a PCA study. So we will not see it in what I say in the software. We'll just go through the slides and then we'll take a PAT case study. So just so far, any questions? If there are any questions, feel free to ask. Nobody interrupted me during the sessions. Either I'm very clear or either I don't make any sense at all. Okay, so I'll just ask some questions for, if I'm not getting any response, I just want to see if I'm being clear enough. Okay. So this question is about how do we decide how many principal components we should have in our model? If to give you a hint, this is the plot, and how would you decide where you are going to stop? Yeah, okay, 3 elbow point. Okay, so everybody is quite attentive. This encourages me to go ahead. Thank you so much for response, guys. So nice. We'll move on. Okay. One more. Yes, yes, Sandeep. It is done in random manner, but we have incorporated some code which will always split data in the same fashion. Means if you are going to import same data second time, then probably you will get very much same training and test data. So this needs to be rechecked, but we have that piece of code inside. So it should happen that if you're importing same set of LCMS data, let's say initially you had sample 1 2 3 in your training set, in the next time also they will be in the training set. This should happen most of the time. Am I clear? Sandeep, did I answer your question?
Participants currently are muted, so kindly write in the chat box.
Okay, thanks Sandeep, thanks for confirmation. Okay, so now we will see application in raw material selection and reverse engineering. For this, we will go through the slides directly. We will not see this in software, because in software there's so much of information and I also get carried away. So to be more focused on topic, and we need to cover one more case study which is for partial least square regression, and for that case study maybe we'll go to the software again. So this is an ongoing work actually in St. John's University, and it's Dr. Ketan Patel's team which is working on this. They're making an orally disintegrating films, like the ones which you administer orally. These are thin films which you apply on your tongue and they get dissolved. So the problem statement here is that when the formulator is trying to make the films in-house in their laboratory, they're very brittle. So they want to find a solution to have um, thin films which are not as brittle, and they are more or less similar to the marketed formulation which is competitor for the same kind of same API. So for example, if you have Wix, you are trying to make an iodex or something similar. So that's why um, uh, they used texture analysis data for building this chemometric model, and I helped them to analyze the data using principal component analysis. This is still ongoing work. You will see a lot of data is masked here, and we have further plans for doing more work in this. So experimental strategy is that they prepared several in-house formulations and innovator formulation, which is not really you can say it's a marketed formulation, and they use marketed formulation and innovator formulation, subjected it to texture analysis study. So in the beginning of the slides when I was mentioning that you need one positive control and you need one negative control, so in this case the positive control is marketed formulation. That is the benchmark which they want to meet. So that is why they generated data also on the marketed formulation. So it's not that easy, guys. We have to work, we have to generate a lot of data. There's tremendous data that this team has generated. I'm just presenting only 1% of what they have done. So this is the correlation matrix which we did not discuss earlier. If you want to read more about correlation matrix, the base how to do PCA in using Data Pandit, and also box plots and other some interesting statistical concepts, then you can visit our blog page. You will see many interesting articles using your data. So right now I'll just is a method when which you use if the variables which you have selected have multicollinearity within themselves. Multicollinearity in the sense when one of your variable increases, the other variable either increases or decreases. So statistically speaking, PCA should be used only if you have this kind of multicollinearity. If you don't have this multicollinearity, there are other approaches which you can use. So, and which are dependent, which need to meet the statistical assumptions which you have need for design of experiments or multiple linear regression. So they have a different set of statistical assumptions that need to be met. So this is about the primary requisite, and I'll explain what this correlation plot means. So it means that you have relationship, Pearson's correlation coefficient for each variable against every other variable here. For example, this is strain with strain. That is why it has a perfect relationship, and it is indicated with red color, which is equal to 1. It is going to change with itself. So that's why it has this one value. It has a perfect relationship, and all diagonal elements have this one value. As you move away from the diagonal, you'll see positive area and strain. So they are positively related, because as you see, this color is somewhere between 0.5 to 1. So if it's between 0.5 to 1, that means if one variable is increasing, the other is increasing too. But if you will see below 0, the colors are blue. So that means in this case, positive area is negatively correlated with initial modulus, because the data shows that if one of them is increasing, the other one is decreasing. So opposite effect is there. So if there is a lot of this interdependence in your data, then you go to principal component analysis as a preferred method of choice. And based on this data, we can see that we have weak because there are some samples which are indicated in green. So this is a weak correlation. So we have weak to high degree of relationship between variables, and there are many which have high degree of relationship. So that is why we can proceed to principal component analysis. Here also we did the same thing. We pre-process the data to bring it onto the same level, and then this is the PCA summary. If we just look at the PCA summary, we see that this is there are 3 groups here basically, and explain variance again here in this case is getting stagnated after principal component 3. So maybe 3 principal components should be enough to explain the model. And here I'll just, because for identifying outliers, we can use this plot here. So this sample 3863 could be an outlier, but I prefer not to call anything an outlier in a commercial or industrial scenario, because if you label something as an outlier without experimental evidences, you may be questioned in future by regulatory agencies. So don't do something which you don't know. Keep that sample, not to have any data integrity related issue. So unless you are very sure, if you know exactly why you should call it outlier, for example, you ended up not processing that sample, that's why it's not this should not be included. If you know something concrete like that, then only remove it from your data set. So initially we see this 3 groups here, but actually if we go here, we see four colors, one for marketed, other one for other 3 are for the in-house formulations. The names are masked. So these are different polymer plasticizer combinations in different ratios, and the formulator experimented with them, and they subjected the data to the texture analysis. When the samples were analyzed and subjected to principal component analysis, we find out that the polymer plasticizer combination 3 here is closer to the marketed formulation. But even if it is closer to the marketed formulation, it has a larger spread. So if it has a larger spread, that means it has a larger variability. If it has a larger variability, we need to reduce that variability so that we will be as good as the marketed formulation. So there is some future work which we need to do to meet the requirement of going to the marketed formulation. But still, this is a good place to start with. So we can select our raw materials. So we had several raw materials, and now here in this case study, I'm just showing you combination of 3. But out of those multiple raw materials, we can see that this one was the closest, and we selected this combination of raw materials for further research. And the other two, the polymer plasticizer combination 2 and polymer plasticizer combination 1 were rejected for future studies. So you go on, if you want to find out cause relationships, you can look at this plot and you can see that the green one, polymer plasticizer combination 1, has higher values for these all properties, whereas the red one, it's not the red one, the yellow one has high values for all these properties, whereas the selected formulation, polymer plasticizer combination 3, has low values for all of these properties, which will be very clear if you look at the biplot, because all the vectors are pointing in the direction of other formulations, and there's no vector which is pointing in the direction of the marketed formulation and the selected formulation. So that is why if we can reduce the value for all these properties by doing some experimentation, we can get closer to our goal. So the takeaway is that by doing a study like this, we can do a bit of reverse engineering to find out what kind of material will fit well for our purpose. Let's say if we are not doing reverse engineering, we can at least find out if the marketed formulation is patented, we can find out alternative materials which we can use for making similar formulation. And if that's not the case, we can use this as a defining criteria for our critical material attributes in 32P2 document. This is related to the pharmaceutical audience. Excuse me. Don't worry too much if you don't get this 32P2 document part. So this is about the
Yes, Rucha, I think you should be able to unmute now.
thank you. So sorry for that disturbance. So chemometrics can be used. There is a question I see now. Is chemometrics can be used for biological assays? Yes, it can be used. Depends on the method which you are using. You'll probably have some, means you'll have multiple columns and you will have one sample related to it. Then you can definitely use chemometrics. So without further delay, I think this is like, do we have time now? I need to check.
We can still take another 10 minutes.
Okay. Okay, then we'll just go to next case study for PAT. This is the problem statement is that this is again a pharmaceutical example. I'm sorry for taking so many pharmaceutical examples, just because I thought since there's more of pharmaceutical audience, I thought of taking these examples. But I'm sure you will find a relevant application in every chemical industry. So in this case also, there is this active pharmaceutical ingredient which gives you therapeutic effect inside your body. And this pharmaceutical ingredient is undergoing transformation when you're processing it. So the hypothesis is that if we use Raman spectra of binary mixtures of these two polymorphic transforms, and we acquire some data, we'll be able to build a model which will differentiate between the two polymorphic forms. We can have a Raman sensor in our manufacturing process, it will determine in real time whether the polymorphic transformation is happening or not happening. So this is the hypothesis behind this study, and this one will take in our software. So I'll just quickly go through most of the steps because now we are very much familiar. Where was that? I think I'm struggling with the internet.
Could be a problem with the server because we are able to see you in the meeting.
Okay, problem with the server. We'll just recheck. Yeah. Okay. So this is partial least square application. We're simply going to upload. We need to upload the data set just like we did it earlier. We will go to this. This is known data. Now, in this case, even if we have acquired Raman spectra, what we have done is we have also acquired results from primary analytical technique. So some kind of technique which is also available in which is a weight chemical analysis uh, in your laboratory. Raman is non-destructive, but this one could be anything. So I'm not sure which technique exactly they have used. You can refer to the article there and you will get to see which technique they have used. So this is the concentration which they determined using primary analytical technique, and this is your Raman spectra. Now in this case we have Raman spectra, so we can go to spectra. Here we can see the spectra. So we have already mean centered and scaled the data. The data is already pre-treated in some other software. So I'm not going into data pre-treatment methods. We have again correlation matrix. This is huge data, guys. So correlation matrix will look a bit different here. And we can see that there is again high because -1 here is blue colored. So there is again high to high relationship between, means the relationship varies a lot from negatively correlated to positively correlated, right from -1 to 1. So this is highly correlated data, and as expected training and test sets got themselves divided. We can visualize here if we want, but we'll not waste much time. We'll go to scores and loadings here. So in case of this partial least square regression, we are building a quantitative model. So if we are building a quantitative model, we should be able to predict what is the percentage of particular polymorphic transform in our sample. So I am just trying to find, I want to see sample names here. So how to interpret this spectra? First of all, here, if we look at this sample number 22, this is my sample number 22. It is closer to component 1. So that means the sample number 22 is influenced most by component 1. And if I look at this component 2, so samples from 13, 14, 15, 16, 17, all these, they are influenced by component 2. How do I know it? If I reduce the size of these sample names, I can see them. There's an option to it. So these are literally 13, 14, 15, 17, and 16. So these are 13 to 16 samples. They are close to component 2. That's why they are influenced by component 2. Another way of analyzing this is we go here, loadings plot. It's the extension of scores plot, means here you will see component 1. Now, because this is a spectroscopic data, it looks like this. If I uncheck this, the loading plot will be different. So how you see how you see this data is dependent on what kind of data you are analyzing. Here, it will just show you component 1 for what for which wavelength component 1 has high response and low response. So this just shows you trend for component 1. So it's not exactly similar to your spectra. It's just that part of, means it has definitely reduced a lot of information in your spectra and taken it into account while building this component 1. So it's not exactly similar to your spectra, but this just indicates trend in which your component 1 is going to change its value. So this is about it. Like we said, RMSEP is very important when we are making quantitative models. So what is your prediction accuracy here? Now we have not used any cross validation method. We can select I want to use CV, and then if I go here, if this CV and adjusted CV are close to each other, that means we have enough sample points and it looks, means the model looks good. If there is a lot of difference in CV and adjusted CV lines, then that means the model is not good. It shows that if with one principal component also we have very low RMACP, which is close to 6, and it is not reducing further a lot. It means it's reducing, but it's not reducing drastically. So this one part is there. You can also look at the R square plot here. So higher the R square, better is the model. So this is definitely for first component itself, it's going above 0.8. So this should be a good model. And again, depending on what level of accuracy you need for your application, you can decide whether you should have this model in place or whether you should not have this model in place. So if we look at the predicted versus actual plot, it gives us an idea about how this model is going to perform. If this is a straight line, and all the models are, all the data points are closely located to the straight line, that means that this is a good model. We just have one sample number 22, which is a bit away from remaining samples. So it may be possible that this could be because of some experimental error, but we are not sure of it. I prefer not to call anything outlier unless I really know the reason. So this is one, how we get a feel of what kind of accuracy we can expect from the model. So if we go to prediction summary, unlike principal component analysis, this one does not show it's going to say this will not have many visual uh, visual plots to see whether you're meeting the criteria or not. Only this predicted versus actual can give you an idea. Here we are going to actually see value for value. So what I mean here is, here the model has predicted 60% form, polymorphic form NaN, I think. So if you want my drawings, I'll go to the table. This, no, sorry, the polymorphic concentration for this NHA labeled niclosamide. So in the prediction summary, it is saying that it has 60% of NHA form and actual is also 60. And also, unlike principal component analysis, in this case, we are making predictions only for test samples in PLS. If you want, we can have a have Outlook, and we can design that part for you. But I think to judge the performance of the model, uh, having an idea about how it performs on test samples is more important. So this is how you can go on comparing. So some cases you will see that there is a big difference. Some cases you will see that there is small difference. So if there is a difference of this 4%, this is something that we should anticipate, because our RMSEP value itself was 6. So because RMSEP was high, we are going to have some variations in the predictions, and we should know how much accuracy we need, because we are going to define our product selection criteria in the beginning of the project itself. So if this accuracy is not enough for us, we might need to get some more samples, we might need to do some more experimentation and make a better model. So this is how we approach it. Once you have your model in place, just like the earlier case, we can save our file here. So, and we can go for predicting the unknown samples. So for predicting the unknown samples, we need to upload another file of unknown data. So we need to upload the model file, which we just downloaded. So I'm just going to upload it. And we need to upload the unknown. This is the unknown. So, okay. It will start making predictions for the unknown samples also. So here we didn't have the primary analytical technique. So unknown samples data. If we go here, it's just the random data. It doesn't have what was the percentage of NHA in the sample. It's not random data. It's actually Raman data. So it will just have Raman data. You just next time when you want to predict whether that polymorphic form is present or not, you simply have to upload this unknown data set. You have to upload your model. Once you upload your model, it will start making predictions in this tab. So first sample. So they have monitored the API during drying process, and they are seeing that as the means these are these spectra are acquired at different time points. So how the NHA value is changing as the process is progressing. So over a period of time, it is reducing, because NHA is probably hydrous form for them, and hydrous form has more water content. So in the beginning of the process, they have higher NHA form, and towards the end of the process, they have lower NHA form. So depending on this process, they can also design a control strategy that if I reach, let's say, 4%, I'm going to stop my process, like this. So this is how we can use data. There are many possibilities if we have that outlook, if we have the passion, and if we are really interested in making database decisions, then there is sky is the limit, literally, because there are many things that we can do with data. So there is this famous quote by Tiffany Boa. She has said that data is the new oil. But data is not oil if you don't know how to make use of that data. You need to subject it to the refinery of analytics. If you analyze it and draw some intelligent insights in the form of gasoline, then only it can become new oil for this century. So this is the takeaway that we should take from this workshop. And with this, I'll close my talk. If you have any questions, feel free to ask me, reach out to me on LinkedIn or any other on my website or any other place. I'll be happy to help you with your objective. Thank you so much for this opportunity.
Yes, thank you so much, Rucha. for your
My
engaging
pleasure.
lecture. Can I request you to stop sharing the slides?
Yeah. Thank you.
Yes. So it was a very engaging lecture, and you have demonstrated the applicability of PCA, SIMCA, PLS using your proprietary software, which I'm sure most of the participants found very interesting. With this, I would now invite questions from the participants. They can unmute themselves. Any questions from the participants? Yeah, probably. I think this was a new topic, so they need some more time for exploring the software. And if maybe if you can also write your email address in the chat box, they can get
Yeah,
back if they have any queries.
sure. I'll drop my email address here.
Yes,
Okay,
thank you.
so my email address is here. I'm also sharing my website. Here they can request access for the software.
Yes. So if anybody would like to connect personally with Rucha, please contact her on the email that she has shared. So once again, I would like to thank both the speakers for being a part of this session. It has been extremely informative. I'm sure our feedback from the participants is going to be productive, and we will share the feedback with you, both the speakers as well, very soon. I am also going to share the feedback form. I request all the participants to kindly fill this feedback form. E-certificates will be shared only on the basis of getting these feedback forms from you all. Yes, so this is a link for sharing the feedback. I request all the participants to fill in the feedback form. I also would like to mention here that unfortunately our third speaker, Dr. Sahu, was unable to engage his talk today. So we are exploring the possibility of having this session in near future. And all the participants that have registered today, we will communicate. If we get a positive reply from him, we will communicate to them personally, and they need not register again or do the payment again. We have the session separately conducted. Right. So once again, thank you to all the participants for their patient listening and for their enthusiastic participation. Thank you again, once again, Rucha and Keshav, for a very informative, engaging, and exciting presentation. So with this, I think we are on time, even without the the third speaker. Probably it was for the best. So we can now close this session. Yeah.
We want to wait till some participants. Please confirm if they have received the link. Participants.
I will reshare the link once again in case any of the participants have not received it or would like to check it again. Yes. All right, so we'll be closing the session now. Have a nice day.
Thank you so much. Thank you so much for this opportunity, and thank you so much for this patient listening by all participants. Thank you so much.
Thank you all.
Titled Applications of Chemometrics for Quality by Design in Pharmaceutical Industry by Ms. Rucha Deshpande. Ms. Rucha is the founder and partner at Let's Excel Analytic Solutions for promoting data science in research.